Hi everyone, this lesson's introduction to the cell cycle. Here you can see two images. On the top left you can see one cell dividing into two, and on the bottom you can see a dog changing over time. Do you notice any patterns in these images? Or anything relating to stability through change? What effect do you think the scale has on each of these processes? And here's what I mean by this. On the top, you're dealing with something microscopic with one cell. On the bottom, you're dealing with a macro object. You're dealing with an entire organism. Is this process going to be different on the microscopic scale than the large scale? Things I want you to think about as we go through this lesson. So we're looking at cell division. Cell division is cells' ability to reproduce. Cells reproduce by dividing. One becomes two. Organisms are going to grow over time through this process. When you see the dog growing, it's not growing because its cells get bigger, but it's growing because it's making more cells through cell division. But how does this happen specifically? Well, in order for a cell to divide, it's going to need to make copies of its DNA. Why? Well, DNA contains the code for building new cells. Specifically, DNA has the blueprint for building proteins, the molecule that makes up the structures inside of the cell. So we need to understand how DNA is structured inside if we're going to understand how it's able to be replicated. When the nucleus is fully formed and a cell is normally doing its thing, DNA is bundled up into a condensed structure called chromatin. Chromatin are just fibers of DNA balled up and compressed so that they can fit inside the nucleus. During cell division, we're going to need to expand this out, and we're going to need to divide the DNA inside the cell. When that happens, the chromatin is going to expand out into a structure called a chromatid. A chromatid is DNA wrapped around proteins called histones and nucleosomes, and oftentimes appears like a stick. Now, chromatids will actually come in sister pairs to form a structure called a chromosome. A chromosome is just two chromatids, and those chromatids are identical, attached together by a structure called the centromere. The primary function of the centromere is just to hold those chromatids together. And a fun fact about biology, just so you know, in biology we always use female pronouns when we're dealing with multiple generations. So phrases such as sister or daughter are always used over father or son. When you have chromatids, those chromatids are what we call homologous. Homologous just means having the same information. Not only are chromatids within one chromosome homologous, but chromosomes can come in homologous pairs as well. Let's look at an example. Here you can see a chromatid, and I'm going to say that the top portion has information A, and the bottom portion is information B. If the purple chromatid is homologous with it, then that too will have A at the top, B at the bottom. And this works for chromosomes as well. This green chromosome has A on the top, B on the bottom. If the purple chromosome is homologous to it, having the same information, it too will have information A on the top, B on the bottom. This will be more important later on. So could you explain the structure and function for each of these levels of DNA in these images? Try it out. What is the structure and function for this structure? How about this one? What is the structure and function of this structure? And last one, what do you think is the structure and function for this structure? Well, you can see on the far left, we have chromosomes, the largest structure, made of two homologous chromatids. In the middle, that is a chromatid zoomed in, and you can see DNA wrapping around histones and nucleosomes. And on the right, you can see chromatin inside the nucleus. So let's think about DNA for all life on Earth. How many chromosomes or how much DNA do you think each different organism has? So let's start with a pea. How much DNA do you think a pea has? Well, a pea has 14 chromosomes. Okay. How about a frog? Frogs have 26, so we have more DNA than the pea. What about a chicken? Yet again, we're getting more and more DNA. A chicken has 78. Now, how about a human? Humans only have 46. That's less than a chicken, but more than a frog. Interesting. What about a chimpanzee and a potato? They both have 48. So reflecting on this, is there any relationship between the number of chromosomes and how complex an organism is? Why or why not? Well, hopefully you've come to the conclusion that no, there is no relationship between how many chromosomes an organism has and how complex it is. A chicken has more DNA than you. A potato and a chimpanzee have more DNA than you. The amount of DNA you have has nothing to do with how complex a structure is. It just deals with the reproductive cycle of the organism or how they evolved. It's not how much DNA you have, it's what that DNA does. Let's take a closer look at human DNA. Here you can see all of our DNA arranged in a picture called a karyotype. A karyotype is just the number and appearance of chromosomes in a cell. The way this picture was procured was all of the chromosome, a picture of the cell was taken, cut out, and chromosomes were lined up into the same sized group. Humans have 46 total chromosomes, and these chromosomes come in 23 pairs. You can see pair 1, pair 2, and pair 3. The ploidy is the number of each chromosome an organism has. So in the case of humans, we have 2 of each chromosome. So we are what's called diploid or diploidy. That's just having 2 of each chromosome, and they are homologous. So chromosomes 1 are homologous to each other, chromosome 2 are homologous to each other, so on and so forth. The number of chromosomes, as you've seen, are different between species, and the ploidy differs as well. We're diploidy, but wheat is actually hexaploid. Rice is diploid like us, and potatoes are tetraploid. Again, nothing to do with how sophisticated the structure is. And just so you know, sex cells, sperm and egg, always have half the genetic material of a normal asexual cell. For humans, our sperm and egg are what we call haploid. They have 1 of each chromosome instead of 2. Why? Well, if you have 46 chromosomes and your mate has 46 chromosomes, that child's going to have too much DNA. But if your sex cells only have half, sperm 23 or egg 23, the offspring will have a perfect 46. So that's enough about DNA. Let's get back to cell division. All living things have a life cycle. Life is born, it matures through time, it gets old, it develops, and spoilers, it dies. Same thing is true for cells. Cells are no exception to this. Cells have a life cycle as well that we call the life cycle. So if we're going to understand how cells divide, we need to understand the cycle of life they live. Here you can see a diagram of that right in the middle. So here is the life cycle of a cell. Notice that the largest part of a cell's life cycle is interphase. Interphase is the phase that makes up most of a life's life cycle, specifically 90% of a cell's life. Now, interphase does have 3 specific growth periods. The first period of interphase is known as G1. During G1, this is right after a new cell has been created, the cell grows, it gets bigger, its cytosol increases. After it's reached a sufficient size, during interphase it will enter the S phase. The S phase is when DNA is synthesized. In a moment we'll see when a cell is created, it is half of the DNA it normally has. It is chromatids instead of chromosomes. During the S phase, those chromatids replicate into chromosomes. After its DNA has been replicated, a cell will then enter the last phase of interphase, G2. During the G2 phase, the cell prepares to divide and it begins to produce its organelles. Why is it producing organelles? Well, we'll see this in a little bit, but when a cell is first created, it is missing many of its organelles. After maturing in G2, a cell then can enter M phase, also known as mitosis. This is the phase when a cell is actively dividing. When one cell divides into 2, it is undergoing mitosis. Why would a cell divide? Well, there are many reasons. One reason is for reproduction. If an asexual cell needs to make a new cell, it's going to divide. This is the primary method of reproduction for unicellular organisms, such as the amoeba that you can see, but this also happens in our body when we need to make more cells. For example, for growth. Here you can see an embryo developing. From fertilized egg to a multicellular organism, we're going to need to make a lot of cells. You're made of trillions of cells. One cell needs to become 2, 2 needs to become 4, and so on. Cell division is also needed for repair and renewal. Some organisms can lose body parts like the tail of a lizard. We are constantly losing our skin. We need to be repairing and replacing those cells that die off, and division is the primary way of doing that. So let's look specifically at M phase, mitosis. During this phase, the primary goal is to divide DNA. By dividing the DNA, we're going to be able to have one cell become two identical daughter cells. This is asexual reproduction, so there's no genetic diversity. Mitosis has 4 phases. Note that I'm not including interphase in mitosis. Interphase is not a phase of mitosis. It makes up 90% of the life cycle. Mitosis begins with its first phase, prophase. After prophase, we enter the phase metaphase. That's then followed by anaphase and ends with telophase. Great way to remember the phases of mitosis is PMAT or IP on the mat to remember the entire cell cycle. IPMAT, interphase, prophase, metaphase, anaphase, telophase. So before I speak about each specific stage, take a look at this animation on the top left. Once it starts with the nucleus, notice what's happening to the DNA. Try and focus on any patterns you see. I know this goes a little fast. You can see it's all together in the middle and then splits apart. So let's start with prophase. Prophase is the first phase of mitosis. This is what a cell looks like when it's undergoing prophase. 3 major events happen during prophase. The first is centrioles appear, and you can see them in the top of the diagram. Centrioles are those cellular structures found only in animal cells that produce spindle fibers. Next, the nuclear envelope dissolves. Notice how the nucleus is a dashed line. Chromatin can fit inside the nucleus, but chromosomes can't, they're too big. So the nucleus needs to be dissolved to make way for DNA to make it out into the cytosol. And the last thing that happens is chromosomes appear. That chromatin is going to begin forming into chromatids and chromosomes. If you were to look at a real cell undergoing prophase, this is what it would look like. You're able to actually see with the microscope all of the chromosomes begin to appear and the nucleus dissolving. After prophase is metaphase. Notice where the chromosomes are now and what happened to the centrioles. During metaphase, centrioles begin to produce fibers called spindle fibers. Those spindle fibers attach to the centromere of the chromosomes and push them to the middle of the cell. And all those chromosomes will align along the middle of the cell along an imaginary line called the metaphase plate. There is no actual plate there, it's just an imaginary line. Here's what you would see under the microscope. You can clearly see the spindle fibers, and all of the chromosomes lined up in the middle. Next up is anaphase, and I'm going to go back to metaphase again. Do you see what happened? All of the chromosomes have been broken apart into sister chromatids. Those chromatids are now going to move to opposite sides of the cell, and that makes it so that each new cell is now getting one homologous chromatid. Each side now has the same information. In this example, each side's getting 2 blue chromatids and 2 pink chromatids. Under the microscope, you can see this very clearly. You can see DNA moving to opposite sides of the cell, and now that middle is completely free from DNA. How do those chromatids actually move? Well, in the middle of the centromere is a structure called a kinetochore. What a kinetochore does is it actually destroys the spindle fibers, and in doing so, that is what's pulling those chromatids to move to opposite sides of the cell. Now, you always want to keep in mind, how are scientists proving claims like this? Well, in this diagram, you can see the experiment that was performed. The scientists in the middle of anaphase broke the spindle fibers close to the spindle and waited to see what would happen. Turned out the chromatids will move all the way to the break and then stop. What does that tell us? That tells us that the spindle fiber is not pulling the chromatids, because otherwise they'd stop moving entirely, but instead the kinetochore is breaking them down, and that movement is what's causing it to move to the opposite side. So now we're at our last phase, telophase. With telophase, you can see now the nucleus is beginning to reform, and the cell membrane is beginning to pinch in. We don't have 2 complete cells yet, though, because they're still sharing cytosol, but we're getting very close. And here it is under the microscope. You can see they're fully moved to the opposite side and beginning to reform their nuclei. Once telophase is passed, we now have cytokinesis. This occurs once the cell membrane is fully pinched in and now separated 2 different cells. Notice we've now started with 1 cell with 4 chromosomes and ended with 2 cells that each have 4 chromatids. So these cells are genetically identical, but what's going to happen now? Well, interphase. Both of these cells are now going to enter G1, where they get a little bigger, S and G2. But notice that we're left with chromatids. we need chromosomes if we're going to have identical cells. We started with 4 chromosomes. Well, that replication is going to happen during S phase. That is why DNA is replicating that point during interphase. So could you explain each phase of the cell cycle? Could you explain an interphase, G1, S, and G2, and then explain how during M phase DNA is split and copied through prophase, metaphase, anaphase, and telophase? Hopefully you can. Now there's a problem that cells can develop in terms of their growth. On the left you can see normal cell division, and then to the right of that, cancerous division. Notice that the cancerous cells continue to grow uncontrollably in number, and they're growing above and below each other instead of growing side by side like a normal cell. That's a problem. How do you think living organisms are going to be able to prevent this problem? Well, the answer to that is also tied to the fact that cells need to be able to regulate their rate of growth. Your cells can't be reproducing at all times. You'll develop too much skin, too much heart tissue, too much brain tissue, so on and so forth. Each type of tissue needs to grow at a particular rate. For example, embryos will divide every 20 minutes, skin cells every 12 to 24 hours, and nerve muscle cells don't really grow at all. So what's regulating this process and determining when cells grow and when they don't? And how could that relate to preventing the problem of uncontrolled division like cancer? Well, cells are regulating their growth in 2 ways. One is using receptors and chemicals to turn growth on, go and divide, or turn growth off, stop reproducing. Another way is cells are constantly being checked at different points in their life. This is to check to make sure the cell is healthy and that all the DNA is free from mutations and should be replicated. So through chemicals and through different points in the cell cycle we call checkpoints is how we're going to do this regulation. Let's start with looking at the chemicals involved with growth. Cells will have a receptor called a cyclin-dependent kinase, or CDK, on their surfaces. If this is activated, it will stimulate the cell to grow. Go do mitosis. Just a vocab point, a kinase is an enzyme that can phosphorylate, so here we're dealing with the transfer of phosphates. The chemical that can activate a CDK is known as a cyclin. Cyclins attach to CDKs, and when they do, that gives the approval for that cell to progress to the next phase of the cell cycle. Go, grow, make it to the next phase. Often, though, cells don't really need to grow. Nerve cells and muscle cells don't need to grow. They're limited in their capacity. It would be bad if they grew too much. And other cells need really specific timing. For example, the cells in your liver only divide once a year. And if cells damage, such as becoming cancerous, we want to stop it from growing before it grows too far. So there is another phase that a cell can be put into that can stop growth in all of these contexts, and it's referred to as G0. G0, or the resting phase, is a phase where a cell no longer divides and stays in whatever phase it's in. It can enter G0 from G1, S, G2, or so on. So to check a cell to either go forward or stop, there are 3 major points we call checkpoints in the cell cycle where that determination is made. The first checkpoint is when a cell is going from G1 to S. Here, the cell's being checked to make sure, should we do DNA synthesis? The DNA in the cell is checked to make sure there's no mutations or anything going wrong. If it's right, awesome. They get some cyclins and move on to the S phase. If not, they could be sent to G0. The next checkpoint is at the end of G2, right before a cell reproduces. At this point, the cell is checked to make sure it has enough cytoplasm, so enough nutrition to divide, and to make sure that S phase occurred accurately, that we have the chromosomes where they need to be with the information the cell requires. And the last checkpoint is the spindle checkpoint that happens in the middle of metaphase. This is a check to make sure that all the chromosomes are properly lined up and splitting correctly. Because if I don't get the exact number of chromatids on either side that I need, a cell can end up with too much DNA or too little DNA. That's the cause of some genetic syndromes, such as Down syndrome. So let's put all of this together. At each checkpoint, the cell is assessed. We're checking to make sure some process has gone through, that it's ready to go to the next phase. If everything's a go, cyclins are increased in concentration, and that gets the cell to the next phase of the cell cycle. If something's off, then the cell is triggered to enter G0, where it's put in stasis and no longer can reproduce. There are other factors at play here as well. Just to go over a couple, one is called density-dependent inhibition. This is where cells will undergo mitosis until they sense cells are surrounding them. Or to put another way, touching another cell turns mitosis off. If a cell finds that there aren't any other cells touching them, they'll turn mitosis on until they're completely covered. This is one way that cells can make sure that they are in an even, flat layer all the way across the surface. Another factor is called anchorage dependence. In this case, attachment to a cell below is required for a cell to start growing. Only if they feel something below them will they then continue to grow up and up. This is how organisms are able to accurately grow cells vertically, whereas density-dependent inhibition is for growing horizontally. So let's go back to cancer. Cancer is just unregulated cell growth. These are cells that are able to rapidly bypass through every cell checkpoint. Normally, cancer is stopped. We experience cancers in our body all the time. But there is a gene called P53. And the way P53 normally works is if it detects any damage or errors in your DNA, it will do one of two things. It'll either send an enzyme to repair that damage so that you then can go forward and reproduce accurately, or if the cell is too damaged and it's beyond repair, P53 will trigger apoptosis, causing the cell to die. Why? Well, it's better for this defunct cell to die than to potentially turn into a cancer. Cancer can grow uncontrollably if there is a failure with P53. If P53 itself becomes damaged, now there's no mechanism for checking that damage and destroying the cell. So instead of being degraded or repaired, that cell that's been damaged now just reproduces again and again and again, and it picks up more errors again and again until it becomes a viable colony of cancer cells. You may have heard the term benign growth or malignant tumor. Just to clarify what that means in terms of cancer, a benign growth is a cluster of abnormal cells that stay in one area. Those, when detected, can be surgically removed, and the recovery rate is very high. A malignant tumor, on the other hand, is a cluster of abnormal cells that are able to detach from where they're located and move to other tissues in the body. This is when cancer becomes dangerous and uncontrollable in the body. So cancer will actually develop if there's 6 key mutations that all occur at the same time. The first one is something needs to trigger unlimited growth. Something needs to turn on the cells wanting to do mitosis again and again and again. Another is it needs to be able to pass all of the checkpoints. P53 needs to be turned off. And it needs to be able to likewise escape apoptosis being triggered by P53 or another gene. It also gains immortality or unlimited divisions. Hopefully you remember from DNA replication that cancer cells have telomerase. They're able to repair the telomeres at the end of their DNA whenever they replicate. That needs to be turned on somehow so that a cell doesn't degrade as it reproduces over time. It also needs to be able to promote blood vessel growth. As cells grow in an area, they need nutrition to be supported and grow, so cancer cells need to be able to turn on that ability so that blood vessels can grow towards them and encourage that growth. And then they also need to be able to overcome anchor and density dependence. They need to be able to grow regardless of if there's anything touching them or not touching them. So what's causing all of these mutations? Well, any substance that causes cancer we refer to as a carcinogen. It's just any substance that can cause these mutations that lead to cancer. These things include UV radiation, chemical exposure, radiation exposure, heat, cigarette smoke, or any drug for that matter, pollution, and age, and genetics. You can have a genetic predisposition for more likely being one who would develop these mutations. So hopefully this presentation was a helpful introduction to you to the cell cycle. We went through interphase, G1, S, and G2, all of the phases of mitosis, PMAT, prophase, metaphase, anaphase, telophase, and cytokinesis. And then we saw how cells regulate that process so that cells can grow at different speeds and what happens when a cancer develops. Thank you, and I'll see you next time.